Bienvenidos al canal del grupo de estudio de hormigas neotropicales. El día de hoy presentaremos la charla The Chronicles of Nylanderia, Integrating Phylogenomics and Undergraduate Training to Overcome a Global Taxonomic Impediment, a cargo de Jason Williams, de la Universidad de Florida. Síguenos para más contenido relacionado con hormigas y no olvides suscribirte. Jason is a scientist who focused on studying the taxonomy and systematic of ants. In his research, he integrates various areas such as morphological study, nano-CT, bioinformatics, and phylogenomics. He demonstrates his passion for insects by having 2 ant species tattooed on his arms. But they are not the only animal he loves, as he lives in the company of 2 lovely cats. Jason believes that by using basic knowledge about life, such as collection-based research, we can gain a predictive understanding of biodiversity, and thus better conserve and manage native biodiversity in the face of climate change.
Okay. All right. So whenever you're ready, I think you should have the ability to share screen.
Yep, here we go.
All right.
All right. Thank you very much for that wonderful introduction. I'm very happy to be here, very excited to share with you all today the past, present, and future of Nylanderia systematics research from my perspective. And I'm calling it the Chronicles of Nylanderia. You'll see why. But it is a sort of long, convoluted taxonomic story behind Nylanderia. And I guess let's just begin. So my slide isn't advancing. There we go. Okay. So my research interests are in systematics, and I am interested in using all sorts of different tools and techniques and resources to understand biodiversity. I'm really interested in especially taxonomic training and how these different tools can also be used for education to train the next generation of taxonomists. And I'm also interested in the practical applications of our taxonomic research and why these things really matter. A few years ago, my collaborators and I were able to secure funding through the U.S. National Science Foundation, advancing revisionary taxonomy and systematics to address the taxonomy of Nylanderia. And through this project, we have various goals in mind, including revising Nylanderia taxonomy globally, defining species boundaries, and describing new species, reconstructing the global evolutionary history of Nylanderia using genome scale data, in our case using ultra-conserved elements, and to resolve taxonomic uncertainty and provide tools for rapid identification and effective management of invasive species. And then one additional goal is to, of course, train the next generation of taxonomists and professional insect identifiers through having them engage actively with the research in the classroom and outside the classroom. To give you a general overview of what I'm going to talk about today, I'm first going to talk about what even is Nylanderia. And then after that, each of these bullet points sort of is tied to a distinct paper, some of which have been published or are about to be published. So we'll go through each of these one at a time, but we'll start again with what is Nylanderia. And so the chronicle begins when Emery first described Nylanderia in 1906. He named it after William Nylander, a Finnish botanist who had collected specimens of Nylanderia vividula, what is now known as Nylanderia vividula, from greenhouses in Helsinki. So Emery first described Nylanderia in 1906, but he described it as a subgenus of Prenolepis. And then in 1925, he determined it to be a subgenus of Paratrechina. In 1936, Wheeler determined Nylanderia to be its own genus, and so he elevated it as such. And then Creighton in 1950 moved it back to subgenus of Paratrechina, and just a year later, Chapman and Kapko elevated it to genus again. Brown in 1973 synonymized it with Paratrechina. Smith in 1979 once again, moved it as a subgenus of Paratrichina. Traeger in 1984 said it was a synonym of Paratrichina. And then finally, in 2010, Lapola et al. Determined Nylanderia to be a genus based on molecular results. This is a simplified cladogram from phylogeny of the Prenolepis genus group. So you'll see Paratrichina and Prenolepis are here. And Nylanderia is a distinct lineage from those two genera. So Nylanderia was determined to be a genus at this point. And it's not surprising that Nylanderia bounced around so much because the morphology, even at the genus level, can be pretty difficult. But here's a timeline. This is adapted from a paper, but I made an updated version of it. The blue line in this figure, the blue dashed line, represents the taxonomic changes in Nylanderia through its whole history. But all of these different genera most of these genera have had some issues with taxonomy over time. The Prenolepis genus group as a group is defined by several morphological characters. There's one that Boudinot et al. Described in 2022 that is this modified abdominal tergite 3 with a dorsoventrally oriented groove for reception of the entire pedial. This is an interesting character that has not come up in the literature before. So this is a new character they discovered. So this is a little sort of area that the pedial can be tucked into under the gaster. But it takes a whole suite of morphological characters to really define Nylanderia morphologically. It's not easy to define Nylanderia morphologically unless you consider many characters at once. So here's just a general list of the characters. One in particular that I discovered back in 2016 when I was working on Prenolepis was this mesosomal sutures deep and complete character. It's the bottom of this bulleted list. And this is a character that separates the genus group into two general clades. There are 3 genera that have a fusion of the mesosomal segments so that there's like a continuous sort of blend between different parts. For example, between the propodium and the mesopleuron, whereas for the 4 other genera, including Nylanderia, have these more discrete separations between the segments. So Nylanderia is a large, near globally distributed genus with 125 currently described species and estimated hundreds more awaiting description. It is notably not very present in the Palearctic, but the rest of the world, it's a very common genus. Its centers of diversity are in the neotropics, Southeast Asia, and Australasia. And there is a whole lot of cryptic morphological diversity that contributes to, as we saw with the genus level classification, but especially the species level classification. And then the genus is perhaps most well known for its invasion biology. In particular, the tawny crazy ant, Nylanderia fulva, is a well-known pest ant species, an invasive species, especially known here in the southeastern US, but also in other parts. When we look at characters for delimiting species, it gets really difficult to untangle interspecific variation from intraspecific variation, because you can have characters that in some cases are very useful, but in other cases are not, such as color. Sometimes color patterns are very useful for species, but other times you might look at a single colony of individuals and see just a wide variation of color within a species, within a colony. So it's very careful to pay attention to the context and to look for multiple characters. So color patterns can be very stark, very distinct. This species on the left is Nylanderia steinheili. There are numerous species that have white meso and metacoxy, or whitened sort of joints or trochanters that can help sometimes. But this character, interestingly, is convergent in Nylanderia. There are many different groups of species where you can find white meso and metacoxy. So this is only one of many characters you might need to consider when looking at the species level. The species on the right I picked to show you today because it has a very distinct color pattern where you don't really see very often, where it has a very darkened head and a yellowish mesosoma, yellow mandibles. Like much of the body is yellow, but the head is dark. And it's very consistent as far as we know in that species to be that color pattern. So color patterns are useful until they're not. It's just one of those things. Another thing we look at is pelosity, so macro-CT and the pubescence patterns. This is a very interesting species, one of the most unique looking Nylanderia species that I know of. And I chose to show it off to you today because it has these really unique kind of pin-like macro-CT that you don't see in other Nylanderia. So from time to time, you'll see these very distinct characters that can be useful. But often in most other Nylanderia species, we're basically just counting hairs or looking at the relative arrangements of them on different body segments. But this one is very distinct in its abundance of these pin-like erect macro-CT. Microsculpture is also something that comes up. Probably most Nylanderia species don't have much microsculpture, but there are some that do. And sometimes you'll find it on different parts, different segments, on the head, sometimes on the abdominal tergite. And in some cases, this microsculpture can give the appearance of a brilliant blue reflection, like a blue iridescence. And in those cases, it's a really pretty color. And the amount of that iridescence can vary within and between species. So sometimes that's a consideration as well as looking for that iridescence. And then finally, I'll talk a little bit about the male genitalia in this section here. Male genitalia are perceived to be useful specifically for telling Nylanderia fulva from Nylanderia pubens, but it's also been applicable in other species as well, of course. Generally, what you see in Nylanderia fulva is a more narrowed tip of the gonostyle, which has been called the paramere in the past, and a lower abundance of macro-CT on the entire gonostyle. Whereas in pubens, they can be much broader, or they tend to be much broader, and they have a more of a fan-like arrangement of long macro-CT. So this has been cited as the only morphological character useful for telling these two species apart. Otherwise, the workers are basically morphologically identical and near impossible to tell apart without the use of the understanding of genitalia or molecular techniques. So we run into various challenges in Nylanderia taxonomy in high species richness and abundance worldwide, limited morphological variation with relatively few easily discretizable worker morphological characters, and very strong overlap in interspecific variation. We also run into this issue with the frequent transport of globetrotting species. These are species that move around the world via human-mediated dispersal, often in things like potted plants, ornamentals, but in other materials as well. And since a number of these species do move around the world, it necessitates an understanding of Nylanderia taxonomy on a global scale to correctly identify species. But the challenge with that is that since there's so many cases where these characters are useful, but sometimes they're not, sometimes you really need to know where something is originally from to come up with an accurate identification. If you have a specimen, if someone gives you a specimen and you don't know where it's from, the morphology might match something from one part of the world or another, and you don't know unless you actually know where it came from, how to identify it. So it's particularly challenging taking in species identification in this genus in a global context. And we need to know who these globetrotting species are for practical reasons, of course. I'll talk more about later, or now. So conceptually, the idea of taxonomic preparedness is something that I covered in a review paper that was published in 2020. And this is a paper where I looked at all of the non-native and invasive Nylanderia records in the literature historically, and in museum collections to get an understanding of who the globetrotters are. What are their identities? What do we currently know about them? Because having a predictive understanding of biodiversity is what really helps us with other areas of research, such as ecology and conservation. Biosecurity is very dependent on correct species identification, and so is integrated pest management. If you're considering something like natural enemies, for example, I like to put this example up here. This is a paper from 2018 that discusses taxonomic preparedness in the context of natural enemies. So this is the samurai wasp pictured here emerging from a case, and it is a biological control agent for the brown marmorated stink bug. So these are reasons why taxonomy is practically important. And so I looked through the literature, I found all of these different records, and I summarized them in this paper. I also included descriptions of their distributions, their biology and natural history, anything known about interactions with other species or plants, other insect species or plants, particularly in the context of plant-sucking insects. And I also summarized their known pest status. And I came up with a list of 20 species that had records outside of their known, what we thought are their native ranges, 5 of which only had dubious or needs verification records. And this included looking at antmaps.org as a resource. And then at least 15 that are basically confirmed known globetrotting Nylanderia species. And from that, I put together what are the most important ones to study immediately. And I came up with these 3 species complexes that I wanted to focus on, specifically the Bourbonica complex from Southeast Asia and Australasia, which includes the species Bourbonica and vega. There's the Falva complex from the neotropics, including Falva and pubens, and then the Guatemalensis complex, including Guatemalensis and Steinheili, also from the neotropics. So these were the 3 species complexes I determined were the most important to study. If we look at Nylanderia bourbonica, it is the most widespread Nylanderia species. It's found basically in the tropics worldwide, and well, not necessarily worldwide, but like in many parts of the world. Its type series is known from Reunion Island from 1886, which tells us that this species has been traveling for a long time, long before we even knew what it was. To study these species, what I wanted to do, these complexes, I wanted to use ultra-conserved elements. And in 2019, I co-authored this paper with Miles Zhang about the use of UCEs for arthropod phylogenomics. And UCEs are these highly conserved genomic loci that consist of a core region that is relatively easy to capture with a probe set because it has low variability across wide taxa. And flanking the core region are these regions of increasing variability that offer utility at a wide range of phylogenetic scales. So the idea was we could capture these and use them in different contexts, looking at Nylanderia as a genus, Nylanderia in the context of the other genera, the closely related genera, and Nylanderia within like the population and species level. They also have high compatibility with other data types, such as SNPs, mitochondrial DNA, because you can actually capture other data types from them. So we've been using these for a while. We've been using the Heim V2 ANTS probe set, which includes 2,524 different UCE loci. And the first UCE paper that I published was on the phylogeny and biogeography of Nylanderia. And I asked the research questions: what are the major clades of Nylanderia and how do they relate to the biogeography? What is the age and biogeographic origin of Nylanderia? How many independent origins are there for the globetrotting lineages? And to answer these questions, I sampled 112 samples worldwide, including 92 Nylanderia, representing about 44 species, 9 of which were the, including the 9 of 15 globetrotting species, and then also 20 outgroups from other genera, including Lasius and other Pronolepis genus group genera. And this first round of UCEs, I did all the wet lab work at the Smithsonian National Museum of Natural History. And I generated this maximum likelihood phylogeny of the genus worldwide, and found that there were these major clades. There's one that's an Indomalayan Holarctic clade, which is relatively, it's mostly the Nearctic material, which I didn't have much representation in the first round. But it's primarily the Nearctic and a couple of like Northern Asian and like Northern Asian species mostly. And then there's a near global clade that includes a large neotropical clade, and then an Eastern Hemisphere clade, basically, which has like an Afrotropical Malagasy subclade, and then various other sorts of Australasian and Asian clades. And then I found that there were at least 5 independent lineages of globetrotting across the entire tree, at least 5. To do dating and biogeography, I used the birth-death model in BEAST2. I used 2 fossil species for fossil calibration, and I used a reduced data set of 44 taxa. And then I did biogeographical analysis in biogeobears. And in this analysis, I found that Nylanderia had an Indomalayan origin about 35 to 45 million years ago. And then from that origin, numerous other clades had arisen, and clades related to geography. There's the Nearctic clade, one of the Australasian clades, an Afrotropical Malagasy clade, and then the Neotropical clade. What was interesting is that the Nearctic clade and the Neotropical clade were distantly related, and implied the 2 independent dispersal events to the Americas from Southeast Asia, to the Neotropics 21 to 32 million years ago, and then to the Nearctic 8 to 22 million years ago. Now I'm going to switch gears, and I'm going to talk about molecular species delimitation. This is the next thing I did with the UCEs. I wanted to see what different sorts of models and data types could tell us about how many species there are in each of the major complexes. So again, looking back at Nylanderia bourbonica and this global distribution, you can ask the question, is this one or multiple species? And to do this, I did more sampling. I did more wet lab work, and came up with 2 data sets. One focused on the bourbonica complex with 50 taxa, and one that focused on the 2 neotropical complexes, the Falva and the Guatemalensis complex, using 38 taxa. And one of the methods applied to the UCE data was allelic phasing. So you can phase each of your samples into 2 haplotypes. And it essentially is doubling your sampling, because you're taking each of your samples and you're dividing it into 2 separate, your maternal and your paternal chromosome. So using the full UCE data set, I was able to take out different data types, including mitochondrial CO1 data, the phased UCE data, and then SNPs data, and applying these different delimitation models to them. I won't go into too much detail about what each model is. I'm just going to go broad strokes and talk about how they compare, but they're kind of summarized here. So each model uses a different data type. And then looking at them across, so on the left column is my initial morphospecies hypotheses before the analysis. The following columns are the different delimitation models. And so we can see there isn't really consensus across the different models in any of the groups. And then the rightmost column is sort of like an arbitrary decision based on an accounting for my a priori hypotheses based on morphology, and then a summary of the other models. So if we look at this in the context of each group, what I have labeled here as Nylanderia burbonica is what basically looks like burbonica that people have thought were burbonica in the past. And these samples turned out to be at least 5 different species when we look at how they should be delimited. So with the phylogenetic evidence, I was able to go back and actually find a couple useful morphological characters for separating some of them out. Very subtle, but you wouldn't know unless you had an idea that they might be different. So basically finding that what we've been calling burbonica might be at least 5 different species. And there are a couple of these that are in China that, perhaps maybe this species its range has been a little bit overestimated because of our lack of understanding of the species boundaries. Some of these are relatively distantly related from what should be called true burbonica based on the type material. And then Vega came out as just a single species. You'll see, of course, these different columns that show the labels of what the different methods determine to be species. Again, many of them tend to oversplit. So I wouldn't really consider them as having the most utility. The one that seemed to be the most useful was using what was called Stacy on the phase SNPs. Those were most consistent with a priori hypotheses and didn't seem to oversplit. If we look at the Fulva complex, none of them grouped any of the samples of Fulva to pubens. I have these labels of CF Fulva here, but later to determine them to be pubens. But none of them grouped any of those samples together as both Fulva and as a single species. So it seems so far that pubens and Fulva are these reciprocally monophyletic species. To conclude this section, basically I made decisions about species boundaries that are most conservative and most congruent across all the methods. However, again, most of the methods oversplit. And then the Stacy method was most consistent overall with the a priori hypotheses. And then in the paper, I discuss how these species boundaries have significance for conservation and invasive species management, because the way we recognize species can have major impacts on policy. Now I'm going to talk about male morphology. So this is a paper I submitted just last week. It's called Picking Up Signals in Male Genital Morphospace and Integrating Phylogenomics to Delimit Neotropical Nylanderia Species. And there are a few challenges we run into with male morphology, particularly in ants. But some of these challenges are in general. With genitalia, there are often these assumed a priori, they're assumed a priori to be informative about species boundaries without rigorous testing. So much of the support for their utility is that somebody saw that genitalia look different between 2 different species or 2 different groups and determined species based on that. But we also run into this issue with frequently there are small sample sizes, especially in ants, which are underrepresented and difficult to associate with conspecific workers unless you have entire nest series or are able to use molecular data to associate a male with a worker of the same species. And then again, these issues with potential misidentification with invasive ants and having morphologically cryptic workers that can be confused with native and endemic species. We need to be able to have more males representative with those workers to be able to have accurate identification, unless we're using genetic tools. So coming back to the Fulva pubens Gona style character, this is again the only morphological character that seems to be useful for separating these 2 species. And so the problem was that relatively few pubens males have really been considered when looking at this character. And I don't think we've really been thinking about the amount of intraspecific variation that could happen, or whether there could be interspecific variation where maybe some males of Nylanderia fulva might have gonostyles that are more shaped like those of pubens. I think a lot of information is still missing. So what I wanted to do was test whether there were we're looking at interspecific differences, or could there be, interspecific variation that's happening here. And so I wanted to ask, are male genitalia informative of species boundaries in Nylanderia? To what extent do intra and interspecific shape variation in genitalia overlap? Are fulva and pubens truly separable as distinct species? And there were 3 fulva CO1 lineages previously identified a couple years ago by Fernandez et al. That I was wondering, could we look at those genitalia and determine whether each of those should be considered distinct species? So for this project, I had 2 different data sets. There was the 2-dimensional geometric morphometrics data set, which included 83 specimens, primarily representing 14 species found across Mesoamerica and southern Mexico. And then a 3D GM data set, including 38 specimens representing the 3 CO1 clades from fulva and from pubens as well across native and non-native ranges. And so this is a collaboration with the Calcaterra Lab and Maria Belen Fernandez, who sent us samples that we could use to perform these analyses. So we have these different CO1 lineages. There's the blue lineage, which includes all known samples of fulva from the southeastern United States. So all of the invasive ones here are from that clade. There's the yellow clade, clade 2, and then the red clade, clade 3. So we received all this material to look at the the gonostyles of these different clades to see whether we could detect differences looking at the genital morphology. For the 2D GM data set, I did a bunch of dissections. Specifically, I looked at the different parts, but specifically I was most focused on the gonostyle, again, because this is the most apparent structure for telling differences in the species. It's the most cited character for such. These are different examples of slide-mounted gonostyles. Each of these represents a different purported species. But if you looked at within species variation for these, there is a whole lot of interspecific variation as well that you could see just qualitatively within each of these different species. For the landmarking, I performed that in TPS Dig 2. I used 3 landmarks and 2 curves, each with 36 semi-landmarks for a total of 75 total landmarks and semi-landmarks. And for the gonostyle, I specifically focused on the most sclerotized part of the harp because there are parts of the gonostyle, and in some species more than others, you might see that they kind of distort a lot more. And so I wanted to focus specifically on something that's not going to distort. So I chose that most sclerotized part of the structure to minimize the amount of error that might be introduced. For the 3D GM analysis, I did nano-CT to scan genital capsules of fulva and puben specimens. I did the reconstruction in dragonfly. And this part of the work, much of it was funded by the Society of Systematic Biologists. And so this is an example of what the genital capsules look like after reconstruction. The gonostyles are segmented out in yellow here. So these are the structures that I did the landmarking on. And so I did the landmarking in the geomorph package in R. I chose 6 landmarks and also applied a mesh of 250 semi-landmarks for this one. And this was covering basically the whole structure, not just the harp. And then all of these analyses, the 2D-GM and the 3D-GM, I performed in Geomorph. Used an ANOVA on centroid size and a MANOVA on shape. I did pairwise comparisons between species using Tukey's HSD, and did a principal component analysis. And I also looked at these in the context of UCE phylogeny, once again. These are phylogenies generated using 236 terminals, representing about 50 species, 26 which are known as known species, and 24 that we considered new based on examination of type material. And so I did these analyses to compare with the GM results. And I found that there were, once again, these two American clades. Only this time, there were representatives in the Nearctic clade that it seemed that they had extended down into Mesoamerica. And in one case, there is at least one species that was found as far south as Argentina. And in that clade, none of the species are known to the Caribbean. And then what I'm now calling the exclusively neotropical clade has no known representatives in North America and at least two distinct radiations into the Caribbean. But we can look at these gonostyle shapes in the context of this phylogeny and ask whether, do these shapes represent also, do they predict clade membership as well? So looking at the morphospace of the 2D GM analysis of the primarily Mesoamerican species, there were significant differences in most cases between species. In some cases, there's heavy overlap and not, we'll see like clear differences in the worker morphology, but like no differences relatively little difference in gonostyle shape. But in most cases, the gonostyles were significantly different in shape. Looking at clade membership, found that shape does predict clade membership with pretty much all of the Nearctic and Mesoamerican species toward the left in morphospace based on the first principal component, and then the exclusively neotropical clade tending towards the right in the plot. When we look at the Fulva complex, UCE phylogeny, I did not recover any of those CO1 clades as monophyletic, but I did still recover pubens as monophyletic. And then if we look at the 3D models of the gonostyles, there is a whole lot of variation of the structure that you can see, but generally pubens tends to be more short and broad, as was previously observed. And then if we look at the morphospace for this complex, we do see significant differences between pubens and fulva in centroid size and morphospace in shape. There was some overlap of the fulva 3 clade with pubens. I think that was only due to small sample size, because if you lump all fulva together and look at centroid size between fulva and pubens, you do find there is statistical significance between the two groups. I think it's just that because there were only had 3 representatives of the fulva 3 clade in the analysis, it did not separate from pubens in centroid size. I wanted to see, however, if adding a third principal component to the morphospace would help us even better draw a line between pubens and fulva, because these principal component scores are kind of low. But I do still think that the analysis does show us something, that we are seeing differences in shape between these two species. But I think we still also need to be very careful in the way we interpret them. So to conclude the male genitalia part of the talk, male genitalia are informative of species boundaries and clade membership. Like other characters, there can be great intra and interspecific variation. And again, we just need to take great care in interpreting these characters. And fulva and pubens gonostyles are statistically significantly different in both centroid size and shape. And the CO1 clades of Fulva do not resolve as monophyletic using the UCEs, and their gonostyles are not statistically significantly different. So currently, I don't feel that we should be calling those clades different species, but I could be, there could be something else in the future that maybe supports them. But I don't think so currently. I don't think we have the supporting evidence to call them different species. But so these results do inform revisionary taxonomy because we have determined at least 24 new species to be described in the neotropics alone. And now I'm going to move on to, well, before I move on, one other thing that I discovered from this work was while doing dissections, I discovered this mutant male Nylanderia fulva, and I wanted to do something with it. So this was accepted earlier this year. It hasn't been published yet, but this is a mutant male genital capsule where I found that there were a few extra structures that had developed. And I had the CT scan it. I really wanted to see it in this sort of model and be able to count the different number of the different gonostyles and the different ocelli. So the yellow are the gonostyles. On one side of the body, they're more than what you'd expect. The left side is relatively normal, but the right side has extra structures there that shouldn't be there. I'm going to move on now talking about the cure course. So this is work that we did in collaboration with a class of students to describe new ant species. So the taxonomic impediment is specifically when talking about training. Training is a big part of what we would consider the impediment, in that education has been changing with less of a focus on taxonomy and organismal biology in curricula, and natural history museums are basically becoming the last bastions of the practice. In 2008, Wheeler described the new taxonomy, where in the past, taxonomy was a small-scale cottage industry, and we should be moving in the present and future towards a more collaborative team sport sort of taxonomy, something like this. So that got us thinking about how can we use cure courses to maybe promote the practice of team taxonomy and begin training and attracting more students toward the career of being a taxonomist. So our cure course is a cure for the taxonomic impediment. So cure courses are lab courses in which students tackle authentic research problems. They are available at most US universities, and they serve as a means of exposure to a large, diverse student body to basically any sort of scientific research topic you want to run a cure on. They also result in real publishable research that students can actually become co-authors on, depending on their level of commitment. And then they also are a means of workforce readiness. They introduce students to career opportunities and give them training. So we figure cures can help us recruit and train students. So in spring 2023, we ran this Nylanderia taxonomy cure, and this is our instructor team here. We ran this between two institutions, University of Florida and Towson University in Maryland. And these two groups of students, 30 students at University of Florida and 15 at Towson, would periodically meet over Zoom, and we would do this interinstitutional collaboration on the work. They were all working on the same research questions, basically looking at species delimitation. But we had this list of learning outcomes related to taxonomy research, and international research, and things like ethics in conducting research, and just like how to collect data and curate data as well. Various different topics related to research. So, and we also gave them the purpose, of course, of this sort of research is important to invasion biology and understanding a taxonomic preparedness. This is why our work is important. So giving them a purpose to this work was also important to us. So the students over the course collected and analyzed morphological data for 174 Mesoamerican Nylanderia specimens from about 25 species. And they were given the task to delimit, describe, and name new species. And the ones that, for any that were able to really commit to the project. They are given co-authorship on the paper. And then at the end of the course, we had a poster symposium where the students had the opportunity to present authentic research to the rest of our department at University of Florida, the entomology and evolutology department. And this is basically the first time any of them had ever made a poster, which was a great learning experience for them. And then 8 of these students came with us to the 2023 Entomological Society of America annual meeting. And we had 4 do oral presentations and 4 do posters. And again, this is their first professional meeting, and 2 of them took home first and second place in their section for their presentation. So these are the 4 new species that they are describing in this paper, or we are describing in this paper collaboratively. In addition to the research itself, we are also able to collect pre and post course data. These are built into CURE courses. So we were able to gauge before and after the course how much the students had an understanding of taxonomy and their ability to explain taxonomic concepts to others, which overall appeared to increase. We were also able to look at interests before and after the course in entomology and taxonomy. I should add that the class was open to non-entomologists as well, and most of our students in the class were not entomology majors. So we could also see before and after the course what the students thought about their interest and relevance to their future careers of these subjects. We did interestingly see that students found that taxonomy wasn't as relevant as they initially thought to their career interests. But this is basically, this CURE course in a way becomes a sort of filtering mechanism. It introduces students to taxonomy as a potential career course or career. And the ones that identify with it, they stick with it. And the ones that learn that taxonomy is not for them, they learn it's not for them. And it's exactly what we want to happen. We want to find the ones that want to be taxonomists. But we need to expose students to that possibility before they can even know that. So we do find some of these students did start to approach us about like how they could continue doing taxonomic research after the course. And so those, we identify those as the systematists. So the number of students that were likely to want to do the research, they wanted to stick with us and keep going. So that kind of allows me to segue into some undergraduate research that's ongoing with additional Nylanderia taxonomy. I've had a student, Jack Williams, who's been working with me since last year. He got a University Scholars Program scholarship to work on a taxonomic revision of a species complex. This is the Ostroxidua complex, which this species was described in 1984 with the caveat that it's probably multiple species, and its range is pretty wide from Utah down to Costa Rica. So Jack looked at the morphology in the context of our UCE data sets to figure out what the species boundaries are. And so this is part of the Nearctic plus Mesoamerican clade that I was talking about earlier. And most of the members of this clade have this fine microsculpture and bluish cuticular iridescence. Jack was able to determine that there are at least 6 new species in this complex. This is based on, again, worker morphology, male genitalia, UCE phylogeny, and looking at their geographical distributions as well. So Jack is currently in the process of getting this paper together. We're trying to submit by the end of August. I have another undergraduate working with me named Corbin Puckett. He also got a USP scholarship. He got it this year, and he is working on Nylanderia of the Galapagos Islands. We have collaborators from Ecuador. Some of you might be here, who sent us material, and Corbin is currently working through that material to help figure out what the species of the Galapagos are, and if there are any, we can describe them. Noe Prouty, they are working on a taxonomic revision of the Bourbonica complex. As I was talking about earlier, Bourbonica is most certainly multiple species as we understand it. So they are working on that alongside our PhD candidate, Ash Punath. And Ash is working on his dissertation right now, which is a systematic revision of Nylanderia in the Asian and Australasian regions. And at the moment, he is finishing up a paper on Vietnam Nylanderia. We have a few new species he's describing, and this is some preliminary phylogeny placing these species together. And we also found that we have a new Paratrichina species from Vietnam that he is working on the description of. But that's not the only thing. We looked at this Nylanderia integra. We found this is interesting because this species is actually not resolving within Nylanderia, and we are determining that it is a Zitania, which is interesting because currently all known Zitania are from the Caribbean and parts of Mesoamerica. So this would be a Southeast Asian record of Zitania, which would be a first. And with that information, that got me thinking about, are there more Asian Zitania? So way back in 2016, I hypothesized the placement of a species in Zitania based on morphology alone. I didn't have enough data to really back it up, enough evidence. But thinking back on it, we should go back and look at some of these things. We really need more molecular data representing more broadly the Prenolepis genus group. So that's kind of the direction moving forward after we finish Nylanderia, is looking back at the revisionary taxonomy of other genera such as Paratrichina and Pseudolasius and some other things. I have some ideas for the genus Prenolepis that I want to go back to as well. So yeah, the Prenolepis genus group, there's still much more work to be done. And with that, that's about all I have. So thank you very much for listening, and I'd be happy to take any questions. Thank you.
Okay, thank you so much, Jason. That was really, really awesome. That was just a lot of information.
Yes.
While we wait for some questions, okay, the recording's back on. Okay, well, while we're waiting for some more questions, I actually have a question, and it is, how did you manage to motivate like the undergrads to actually do this endeavor? Because I've tried with not with Nylanderia, which is a challenge in and of itself, but I've tried to motivate students into the usefulness of taxonomy and those kinds of things, but it's difficult, like especially nowadays, as you were saying, like it's hard. Like taxonomy's fallen off the main scientific kind of like zeitgeist. So yeah, how did you get so many undergrads to get interested in this?
It is very difficult, and I think it's part advertising. It's part that we have such a big instructor team with very enthusiastic people that are very effective at getting students interested in taxonomy. I mostly represent more of the research side, so I try to bring I think I do bring a good amount of enthusiasm, but I have to credit a lot of our other instructors with really helping with that. And part of it too is just finding the students. It's not easy, but if you open up the possibility of them taking a cure course in taxonomy and then advertising it well across the university, then you do get some hits. So it's finding the ones that stick around, that want to keep doing the research. You have to really put it out there. You have to put it out there as a possibility.
Okay. All right, let's run down some of the comments. So Luis Calcaterra says, nice talk, Jason. Congratulations for the enorme trabajo. Latke says, wonderful stuff, especially heartening is exposing potential new talent to taxonomy, which agreed, it's really important that we get new taxonomists. Claudia Xavier says, awesome work. Congratulations, Jason. It's the way to see if we get any more questions. I think people are still processing the amount of stuff that you talked about.
Thank you.
Okay, so Gabriel Cardoso says, congrats on the lecture, and asks, is the CURE program for taxonomic courses open for international students?
That's a good question. So the CURE model is something that is very adaptable, and I'm not sure, I don't have a whole lot of knowledge about CUREs internationally. I know here in the US, we have a lot of our universities have a CURE program where you can develop your course and offer it to students. There are people who develop cures that you can rerun over and over again. And some people develop cures that other people can pick up and run. I don't think anything currently exists for taxonomic training. I think we are kind of like among the first people to run a taxonomy cure like this. So I wouldn't say we don't really currently have anything open currently to international, but it's a possibility. And if it's something you're interested in, if you're working as an instructor at a university, like I said, I don't know how much of this is available internationally, but it is a possibility to develop your own cure and run it. We could absolutely share that syllabus.
Yeah, you saw
Jennifer.
Following question. Yeah, that would be very useful. Because I also have where I'm at, we also have implemented a few cured programs, but yeah, they are a very American like institution kind of focused thing. The syllabus can be applied anywhere. So
yes,
especially since you've already developed this taxonomic one that's very expansive and all of the tools of taxonomy, it would be very useful to apply all over the world if we can. Wonderful.
Yeah, and our cure was very morphology focused, but you can develop a cure for doing molecular work or bioinformatics if you can find the students interested in doing that sort of thing. Cures can be very flexible.
The person that's talking in the talk will be available. Yes, we recorded the talk, and it will be uploaded onto our YouTube channel soon. Okay. So Gabriel is asking, I think I wasn't clear. Sorry, I'm a student that really wants more taxonomic education. I want to know if in UF you're open to international students.
You're asking if our cure is at UF. So we don't currently have another taxonomy cure plans at the moment. It is something as a possibility in the future. I don't know. I really don't know off the top of my head if it would just be open to international students. I imagine it could be. But I do have more ideas for running cures, whether it's here at University of Florida or if I find a job somewhere else. I would like to keep working with cures to do other things like more general collections-based training with students. But cures can be anything.
And Jennifer Hiron asks, with the Mesoamerican species, are you collaborating with local scientists?
Yes, for the Mesoamerican work, it's primarily based on collections already made by Jack Longino and others through the Llama and ADMAC projects. I don't have any specific scientists I'm currently collaborating with on that material. It's very extensive material, but I don't currently have any collaborators, at least in terms of publishing with them.
Okay. So I'm assuming you're very open. Currently I'm collaborating.
Yes, absolutely. If anyone wants to collaborate on Nylanderia, absolutely. Yeah, we currently have this paper in submission with the Calcaterra lab. the Galapagos work, as you just mentioned. I'm totally open to collaborating.
Okay. And Claudia says, will you still work with nano-CT? I've just started doing it with spiders, and it's a whole new world to explore. I'm going crazy. It's very exciting.
Yeah, I love nano-CT. I'm absolutely going to keep doing nano-CT projects. I don't currently have a new one planned yet, but I've mostly focused on the sclerotized parts. I know, of course, people like Adrian Richter last week or a couple weeks ago. He focuses on like musculature as well. But a lot of the specimens I've scanned so far for the Nylanderia work, a lot of them are really old too, so you don't really get muscle. There were a couple newer specimens, even though that I dry preserved, I was still able to get musculature for it. But again, I was mostly focused on cuticle. But yeah, I'm really open to the world of doing more nano-CT.
Okay. Okay, I think let's wait until 4:10. And looks like there's a hand up. So what just shows up. Yeah, if you want, you can go ahead and talk.
Hi, I don't know if you can see me.
Yes.
Okay, cool. Hi, Jason.
Hey.
I joined a little late. Sorry, I got the message with the link. I saw it a little late on the. I missed most of it. So the talk is shared. That's awesome. So I'll watch it again. I wanted to say that I didn't contact you. Sorry, I forgot that I was going to be out of town. I just came back from Austin with a group to work with a group over there, Dr. Plaus, in the invasive species that works with the phorid flies and invasive ants. They are working with the Nylanderia there. And I have some more fresher specimens of Galapagos and also the coast, I believe. So if you are still interested and would like to add it to that study, we can arrange when they are still mixed with other ants. So I have not had the time to separate them.
Oh, that's fine. I can give them to Corbin and he can separate them. He'd be very excited for something like that. Definitely, the more the merrier.
Okay. The only thing I will ask is they are, because they are in the molecular ethanol and stuff, try to be a little careful. But anyhow, we can talk about that.
Absolutely. Yeah, thank you.
Thank you. Great talk. I was able to see. Awesome. And I saw the posters. I think that's the class you are presenting now from that.
Yes.
That was awesome. Yeah.
Yes.
I really enjoy watching those posters. Very cool. Thank you.
Thank you.
Okay. All right. I think we're gonna close up. So again, thank you so much, Jason, for giving us this amazing talk.
Yes, it's my pleasure. Thank you so much.
Yes. So again, Gabriel saying thank you. It was a true pleasure. And yeah, I'm really glad. I'm excited to steal some of these ideas.
Yeah, if anyone else has any other questions or Jean-Pierre, you have any questions, just shoot me an email. I don't know if did I give my email? I don't know. But yeah, I'm available if anyone ever wants to know anything. Nylanderia.
All right. All right. Awesome. All right.
Thank you.
Listo. Entonces, con esto cerramos nuestro encuentro micológico. Como ya mencionamos, lo subiremos a la página de YouTube dentro de unas semanas. Y muchas gracias a todos por asistir.